This video is cytoskeleton actin, actin binding proteins, and the cell cortex. I wanted to warn you that the way that I wrote out the vocabulary was a little bit different this time. So basically there's the actin binding proteins, and then there's a whole bunch of different types of actin binding proteins which are listed here, and instead of writing proteins for every single one of them, I just put the name and then put proteins at the very end. And just as a reminder, we've seen the severing protein in the Inner Life of the Cell video. So the Inner Life of the Cell video, the cytoskeleton is featured quite prominently, and this particular version of it, the full version narrated, is really great to kind of check out, starting at 1 minute and 50 seconds and then going until 4 minutes and 25 seconds. Some of the things that I think this video highlights, which are really hard to capture in a textbook or a lecture, include the actin filaments. And here they're showing the cross-linking. So we've got a long strand that's going into the figure here, and then another that's going perpendicular in this way. So shorter one in this aqua color and longer one in purple. Those actin strands aren't really different from each other. They're just highlighting them in different colors so you can see that they're cross-linked. And so that is found in certain structures of the cell, and other locations which they mention in the video would be, for example, deeper within the cytoplasm. Then we could see a different organization of the actin filaments, and that there would be actin binding proteins that would facilitate it. So in this kind of like yellowish kind of crookneck thing, that's an actin binding protein. And I really wish they would have put this title in there, but I just typed it on top of the screenshot. So there you go. At this point, they also mentioned that the actin is kind of gel-like in this region, and so we're going to hear about that in some other cases. So that's where it's found at really high concentrations, and so it's going to facilitate some of the processes and remodeling of actin, which is essential for core functions of the cell, including motility. This is figure 17-30 in your book, and basically what we're going to do is check out a whole bunch of different actin binding proteins. But let's start off at the top of the figure here. So basically inside the cell there's tons of actin, both as a monomer as well as a polymer in its filament form. And usually these filaments are cross-linked. There's a variety of different ways they can be cross-linked, and so that's where these binding proteins come in. Moreover, it's unlikely that there will be individual actin filaments. Remember that actin filaments are very flexible, but they're not as stable, thick, nor do they have as much tensile strength as some of the other filaments. And individual filaments are unlikely to be found, whereas it's quite common to find the filaments cross-linked either as bundles or networks. So let's go through and talk about the actin binding proteins. First off, you do not need to keep track of the side binding protein, but we will talk about the rest of them. The actin binding proteins are shown in green, and so each of them has a different role and has a different protein structure and sequence. However, they're shown in just in green for all of them. So actin binding proteins will bind to actin and modify its properties. And the first one that we'll check out is the monomer sequestering protein. So that's up here. And so zooming in, you can see that there are little actin monomers, which look like that kind of little nub, and then there are, which are in red, for the actin monomer, and then the monomer sequestering protein is shown in green. And the monomer sequestering protein, this is the reason why the actin does not just spontaneously form filaments constantly. So monomer sequestering proteins allowed there to be this free kind of reserve of building materials of actin monomers. And then depending on what other actin binding proteins are present, then those building units can be put together in a variety of different ways. And next up, let's talk about the bundling proteins. So we move over here to the right. So bundling proteins are going to hold together actin in parallel. So the actin filaments are held together in parallel to one another. And this is much like how a bundle of sticks would be held together. If you're going to carry sticks, you put them all in the same direction. Otherwise, it'd be pretty hard to carry them. And this is a common structure of the actin in filopodia, which are used in motility. And so here we see these long actin filaments, and the bundling proteins are found at regular intervals, connecting one filament to the next. Third is the myosin motor proteins, which we see over here. Again, they are in green. Myosin motor proteins are going to move actin filaments with respect to one another. So one actin filament gets moved one way, and the other actin filament gets moved the opposite way. This is part of the muscle contraction apparatus, as is drawn here. Myosin motor proteins are also found in other sorts of contraction events, including in contractile rings, which are involved in cytokinesis. This is at a smaller scale, though, than you would see in the muscle cells. And the myosin motor proteins can also be used in motility. Okay, let's talk about the capping proteins. So let's call these actin capping proteins, just to remember what we're talking about, because we'll have other capping proteins we'll see later. And those are shown here. Again, it's this little green blob here. And what they're showing you is specifically a plus end blocking protein. You can't tell that from this drawing here. But basically, the capping proteins are going to block whatever would happen at the given end of the filament. If it were a plus end blocking protein, then that's likely to prevent polymerization, because that's where the highest rates of polymerization are taking place. And if instead there was an actin capping protein at the minus end, which would be obviously the opposite end in this case, then it would be most likely to prevent depolymerization, since there's a higher rate of depolymerization at the minus end than at the plus end. Next, there are actin cross-linking proteins, and these will create a gel-like meshwork. These are, for example, found in the cell cortex, and you can see here that there's these little kind of, boomerang-shaped proteins. That's the actin cross-linking protein, and we called these attachment proteins when we saw these in the cell cortex previously. Also notice that I put actin in front of the name here to help remember that it is specifically regarding actin rather than a different cytoskeleton filament. Next is to talk about the severing protein. And so severing proteins, again, are shown in green. They have like this little kind of notch. It looks like it might be a blade. And in the inner life of the cell, these were shown kind of like nunchucks, and they were binding to long actin filaments and then severing them. And basically the idea is that a severing protein would physically remove that association between the monomers of actin and decrease the length of the filament. This can be used in cases where the cell cortex is being converted from a very rigid structure into a more fluid network. And again, this provides a way to destabilize the actin filament structures as another example of looking at regulatory proteins. We want to have things that can help stabilize but also can destabilize. And last, let's talk about the nucleating protein, again shown in green. And so this is binding to one end of actin. Nucleating protein, this provides a site at which the actin filaments begin, and basically what this is doing is facilitating actin polymerization, so that if we had rewound in time, this filament would have been dismantled, and we would have had just an actin monomer or two, and then this would have been added to the side. To remind you a little bit about the cell cortex, here's a figure that we have used in an earlier part of the course. This is figure 11-29, and this little throwback about the cell cortex is to remind you about the difference between spectrin and actin. So at first glance, this red long stuff looks like actin. Remember though that actin can be a variety of different lengths. The red here is actually spectrin, which is a long dimeric protein. So each of these long stretches is one of the subunits, and then they just draw actin in kind of a boring blob way, and it's green here, which is a little bit annoying, but remember that actin sometimes makes very long filaments and other times makes shorter ones. And what's shown here in blue as attachment proteins, that's what we were talking about regarding these cross-linking proteins. And they're actin cross-linking proteins. As a reminder, this is the cell cortex of a red blood cell, and while this is pretty complicated and there's a lot of parts, that's actually considered to be a less complex cell cortex than a lot of other cell types. And once again, that inner life of the cell video highlights some of this appearance. So these little triangles here with the spectrin, that's what we see here as part of the cell cortex.